Hi everyone, my name's Rob. I'm a research officer from the Institute for Molecular Bioscience and jointly affiliated with the Australian Institute for Bioengineering and Nanotechnology at the University of Queensland. And I'm just going to give a brief overview of the work that we're doing towards targeting metastasis or the spreading of cancer cells throughout the body to distal sites. And specifically what we're doing is trying to understand the mechanisms that allow cells to move in challenging spaces. So the keyword is challenging spaces, and the reason I bring this up or emphasize this is because within our body, cells are packed within very, very confined environments. And in order to navigate these environments, it is very challenging for cells. So this is a IHC section or tissue section of lung tissue that contains a metastatic melanoma that is stained in purple brown, as you can see in the middle. And hopefully what you can appreciate is that the cells, both normal and cancerous, are present in a very tightly packed environment. So it doesn't really tell us how they've gotten there and what they're going to do next. And so that's where our work comes in. And what we do is we focus on the dynamics of this process using advanced 3D models. So in this video that I'm about to play in orange, you can see a metastatic melanoma cell, and it's been embedded in a 3D network or a hydrogel that we've made of collagen 1, which is the most abundant extracellular matrix component within the cell body. And when we watch this cell invade within this 3D hydrogel over 5 to 8 hours, what you can see is that it very efficiently and dynamically does so, and it's able to overcome physical obstacles by contorting the cell body and squeezing through very, very small, challenging spaces. Now, the important thing about this is that we don't really understand much of how cells actually do this. And the reason is because the way that we've studied this behavior has been largely done in vitro in very, very different environments. So we initially started by looking at the processes that we do understand and the things that allow cells to move predominantly on in vitro settings of glass, stiff substrates where most of our experiments have been done for the past 5 decades. And one of the things that we know really well that drives cell movement is the microtubule cytoskeleton, as well as other components of the cytoskeleton. So microtubules are these long protofilaments that are composed of alpha and beta subunits. And in this schematic here on the left, what you can see is that these subunits undergo this process called dynamic instability. And they grow and they shrink and undergo these periods of stability in between. And if I play the movie of a cell that expresses fluorescent microtubule markers, what you can see is that these individual long protofilaments grow and shrink in a process of dynamic instability over the time course of minutes. And within cells over the time course of hours, you can see that this dynamic instability of microtubules contributes to cell motility on these hard, rigid glass substrates. So there's also a whole bunch of proteins that regulate microtubule dynamics. And these proteins are called microtubule associated proteins. So for the project that I'll talk about today, the protein that we focus on is called CLASP. And it's got a lot of very well-defined functions that regulate microtubules. And it predominantly works to stop microtubules from shrinking or basically destabilizing themselves. So the way that CLASP does this is through a protein called EB1, and EB1 is a master regulator of microtubule dynamics. So it associates here at the very fast growing end of microtubules called the plus end, and then it recruits CLASP, the protein that I spoke about just before. And in doing so, it stabilizes microtubules from depolymerizing. And so what you can see here in the movie on the left is EV1, the master regulator of microtubules, and you see it growing very, very quickly on the fast growing ends of microtubules. And on the bottom here, you can see clasp in white and microtubules in red. And you can see how similar the localization of these two proteins are, and it's because that these proteins are dependent on each other to recruit to microtubule. At least that's what is known from the literature. So, like I said, a clasp is a very, very potent microtubule rescue factor, and it basically prevents microtubules from depolymerizing, and also has a whole suite of different characterized cellular functions. However, the reason why we were initially very interested in class was because around 2018 to 2020, Anna Akmanova's lab in the Netherlands showed that there were these new functions of clasp, so non-canonical functions of clasp, and they basically showed this in vitro artificial settings where they grew microtubules against substrates. So this is a substrate here, and they're growing an individual microtubule here, and you can see as it butts up against this solid substrate, it can no longer grow anymore, and it basically shrinks over the time period of about 10 minutes. Now, when you grow individual microtubules with clasp, as you can see here, it allows the microtubules to bend and buckle or undergo this process called flexural rigidity, where they're both simultaneously more flexible and rigid. And what was even more exciting was that when you damaged an individual microtubule with a high-powered laser, in the absence of clasp, this individual microtubule fractured. But with microtubules with clasp, clasp is able to repair and heal these microtubules. So we initially thought that potentially what could be happening in cancer cells is that if they do express this protein and they upregulate this protein, perhaps it's allowing them to sustain more mechanical forces through the dynamic and ad hoc sort of ability to repair microtubules as cancer cells invade in 3D. And so that's exactly what we did as our first preliminary experiment, which was to grow metastatic melanoma cells. And what you can see here is a spheroid of metastatic melanoma cells. And as I play the movie, you'll see over 24 hours, these cancer cells invade into the 3D hydrogels that sort of surround these spheroids here. Now, when we deplete clasp, you can see quite significantly, it abolishes the ability for these metastatic melanoma cells to invade into the hydrogels that surround. So this was a really interesting finding to us that sort of suggested or even validated that class could be important as a invasive sort of factor or a protein that enables cancer cells to invade and metastasize. So we looked at a little bit higher resolution, and single melanoma cells depleted of clasp and embedded within this tight, sort of dense 3D collagen network, as you can see here on the top panel. And you can see as we sort of play the movie, these cells are largely caged within the 3D hydrogels here. So when we sort of expanded the pores within the collagen hydrogels and loosened them and allowed cells a little bit more sort of flexibility to move, what you can see as the same class knockdown cell tries to move within this reticulated network is that it actually fragments and dies in the process of trying to invade. So we thought this was a very, very interesting phenotype that potentially showed that these cells depleted of class were more fragile. And so to test this, what we actually did was we designed a whole suite of different microfabricated 3D models that allowed us to test invasion through confined settings. So the one that I'll show today is this circular array, which is an array of circles that basically the cells or the cancer cells have to sort of invade through. And as you can see here, as we put the melanoma cells through, they will effectively navigate these circular pillars by contorting their cell bodies, similar to that first movie that I showed you. Now, the question really for us was how well these microfabricated systems recapitulate what we saw in 3D collagen hydrogels. So again, with the control cells, we put them through, and you can see they quite nicely navigate these micropillars, and they squeeze and contort their cells' bodies through. So in white here, we have microtubules. The nuclei are marked in magenta. And quite sort of satisfyingly, what happens when we deplete clasps in metastatic melanoma cells is that you can see the same cells exhibit a very similar phenotype to the 3D collagen. So they rupture and die, which supported our hypothesis that, this protein class could mechanically allow cells to undergo more sort of mechanical forces as they engage in invasion and metastasis. And so really to really directly test this, what we've done is try to use a system where we can directly apply mechanical forces to cells and see how they respond to that. And so this is a system that we've designed that's based on a similar device from the Peel lab in France. And it's basically a piston with microfabricated pillars that allow us to control the compression height of cells. So in here, you can see cells that are uncompressed in the top-down view and also the side view. And then we can actually fabricate pillars of different heights that allow us to basically compress or squash cells. And the way we do this is using a large sort of syringe that is driven by this piston and locked in place by a large liposuction locking device. And you can see as I draw back the piston of the syringe, this deploys the piston and thereby squashing the cells. And so this is fabricated using inert silicon. And so when we do this and we perform these experiments, what you can see is that the control cells are largely able to sustain these compressive forces. And you can see the microtubules in black here, still sort of persisting over the sort of duration, quite a few hours after we compress. But the class depleted cells, their microtubules are much more fragile, and they sort of disassemble over time. And then we can also see that the membrane ruptures when we use a dye that's sort of permeable to breaks in the membrane, called propydium iodide. You can see that here in the quantitation. So in trying to get at the direct mechanism as to what exactly is happening, we did the same sort of experiments, but using a CRISPR knock-in to basically label clasp endogenously. And this is what clasps look like. This is endogenous clasp labeled with a green fluorescent protein. And you can see it localized to the fast-growing ends. Now, what was really surprising to us was that when we compressed cells or subjected them to mechanical forces, you can see that clasps go from this punctate pattern to a stable sort of localization on microtubules, suggesting that it's responsive to forces. And this is actually reversible. So when we remove the compression device, it reverts to the original sort of dynamics and localization, suggesting that this is a protein that is mechanically responsive and allows cells to sort of stabilize themselves in response to mechanical forces. So one of the questions that still remains from the work that we've done and published is what exactly happens to these cancer cells as we deplete clasp and they invade or try to invade through these confined settings and die. So our preliminary sort of evidence has shown that as these sort of class depleted cells die, they release pro-inflammatory cytokines. And one of the outstanding questions really is, does this recruit and train the immune system to try to recognize these cancer cells in the body? And this is sort of what we're working on here. And as you can see here, these are some immune cells, some neutrophil-like cells that we've begun to culture and try to see whether at least the innate immune system is recruited to these cancer cells. So in summary, what I've sort of shown today is we've sort of identified a role for this protein class, and specifically in the setting of confined migration that allows cells to sustain mechanical forces during invasion and metastasis. And this is a mechanism that is microtubule dependent that allows cells to resist forces, specifically in these settings. And so, like I said, one of the questions we're working towards now is trying to understand does mechanically sensitizing these invading cancer cells result in the recruitment and training of the immune system to further recognize these cancer cells, and if so, how? So with that, I just want to thank all the people that have been involved in the project, especially my PhD supervisors and all of the funding sources. And even though I can't be sort of in person live today, I'm more than happy to have a discussion through email or Zoom at a later date. So thank you for your time, and thank you to all the generous funding sources that have supported this work.